actually look like your typical roly-poly isopod. And then there's also the gnathed isopods, which resemble more of a mosquito um, kind of situation where it has this sort of rotund belly that will fill up with the blood that it's taken from its host. It doesn't look like an isopod much at all. It's very modified to be a parasite.
All right, come on.
This feels like a crinoid seamount. There's so
It,
many.
it definitely is a crinoid seamount. The last watch has been seeing mostly crinoids and a few echinoderms, similar, few other echinoderms, a couple of holothurians and some fish. But that's pretty much the inhabitants of this area. It seems like throughout the dives we've been doing around Palau, there's been a lot of this, a lot of organisms that seem to find their particular niche in a particular seamount or current regime combination, where we see kind of one family or one type of organism that seems to be the most prevalent.
Yeah, I've noticed that too across a lot of the seamounts that I've worked on, that you'll notice there's like one group.
What is just below?
Yeah, there's like a darker black.
It looks like it might have been an
like a
egg.
skate egg.
Yeah, it looked like a skate egg, like the one we collected before. Could we get a zoom on that? Yeah. There. We collected a skate egg on the previous cruise, which was very exciting for one of our scientists aboard who's interested in eggs. There's a little shrimp.
Oh, it's a piece of wood.
It's a piece of wood. Just perfectly shaped like an egg.
Yeah.
Very misleading.
It was the right color and everything.
Exactly. Could we zoom even more? Often on the wood we get some interesting life. Let's see what's going on here. Just looks like a little polychaete maybe.
Yeah, maybe a small brittle star or something. That white thing.
Nice. All right. I think we're good. Thank you. I got very excited there for a second. two shrimp.
Shrimp.
So the Palauan word of the day yesterday was shrimp, and in Palauan, shrimp is ur.
Can you say it again?
Ur.
Ur.
Yeah, you got it.
Oh, cool.
And funnily, I don't know why, it's also the word for laugh.
That's funny. Shrimps are having a good time.
They are, you can tell.
They do look like they have a good time.
Is this a different crinoid we're seeing?
Oh, it's the same one.
It's just from the back.
Yeah, from the other direction.
Interesting. There's a long stalked crinoid right coming into view now.
Yeah, I got the red Proisocrinus ruberimus, and then the ones with the legs, which are the metacrinus.
There's a sponge. And another sponge coming up on the starboard side.
Can we zoom on that white stalk?
Yeah, let's go for it.
Yeah.
What's it going to be?
Looks like there's something
Yeah, there's a little
Some-
brittle star on it.
Push in there.
associate.
It's a Candidella gigantea.
Gigantea.
Mm-hmm.
Oh, where's it going?
Oh, it's moving. Those whip corals, they'll just
They just
they'll just
whip around.
rotate. All right. Well, that's what I wanted.
Okay.
I wasn't sure if it was a bamboo coral or a primnoid, and now I know.
Okay.
It is a primnoid. Oh, there's definitely a shrimp in that sponge.
Yeah, there is.
Oh. Yeah.
Oh no, it hid from us.
I saw it at the
Yeah,
bottom of there.
it was like, oh,
There it is.
oh, there is.
Push in there. Oh, sliding off.
Yeah.
All right, come on.
Well, there's often shrimps that lives in those sponges. They're hard to spot. They're the same color as the sponge. But you can see it in there. Jake's gonna go for the cookie. These cookie stars have a very satisfying shape.
Bad landing. Right, push in there.
How fun it would be if we had star-shaped cookies for snack time.
Whoa.
So this is a sea star in the family Goniasteridea, which we call the cookie stars. That's great, thank you.
Okay, come on.
Is it because of it's a cookie cutter shape?
Yeah, it's like the
Oh.
perfect cookie
It's a
shape.
perfect star.
Perfect star. Yeah, they're all very, very star-like.
In the best way possible.
Indeed, exactly. Bridge, Nav. Can we make a 30-meter move, 225? Thanks.
Fish.
It's a halosaur. You can tell by the way it's swimming. I don't always need to see them up close to know what they are.
Push in there.
Is it the whip of the tail?
Yeah, that whip of the tail and the way it kind of floats.
Near
and
the
how
bottom.
yeah, and it's skinny. They often like float head down.
Are these the ones that would also sometimes be in the perpendicular position to the... Like I think we saw one earlier on another dive where his head was, he was directly upside down. Or it was
Yeah,
directly upside down.
sometimes they do that. They usually are a little bit on an angle. There's an umbellula.
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 What a fun name.
It is really fun. I like saying umbellula.
This is something right there.
So May, what happens if we make it to the top of the mountain?
So if we make it to the top of the mountain and it's interesting, we're going to look around. And if we feel like we've characterized this area pretty well, we may be ending the dive early, is what I've been told.
Okay.
So we don't need to rush up the mountain, but we can do our normal zooms and keep our eyes out for anything of interest to sample or get imagery. And on the way up, we'll do our eDNA samples as well.
Yeah, we're making pretty good time. We have like maybe 670 meters to the top, which we easily did last watch.
Yeah.
Looks
So,
like a bit of a snow globe in the upper.
so is all this rock manganese encrusted? Is that what all the little
Yep,
like
the botryoidal
botryoidal
texture.
textures?
The grapes.
Yes, I was thinking that earlier too. Botryoidal.
Yeah, it's Bacchus.
So Bacchus, is that not the name of the Greek god?
It's the Greek god of wine.
Yes.
Yeah,
Yeah.
and referring to grapes, which is the same Greek. The Greek word for that is of the same origin.
Right.
So
Is this
very,
marine snow?
Yep.
yeah, a lot of. It's a blizzard.
Yeah, sometimes if the sediment rate is too high, you're going to have a hard time finding corals and sponges because they'll get, you know, too dusted, and that's
Fish.
not good for them either.
Mm.
So you're trying to find
Pushing
that
there.
balance
A
between
fish.
high current and enough food. That's another little Basazetus.
All right. Come on. Wonder if it'd be better with downlights off with the rain snow.
Oh, yeah, that does make it look a little nicer.
Yeah.
Just don't see as much in the lower.
Don't turn your high beams on in the snow. Another little fish. I'm at the end of my leash, though.
I'm coming as fast as I can, Jake.
As
Maybe
fast
not
as
as I
you
can.
can.
As fast as reasonable.
Push in there. Got a little halosaur? A little tiny one?
Yeah, a little halosaur.
Tiny.
Well, they have to start small before they can get big.
The previous watch got lucky and collected a couple of fish, so that made Jeff very
All right,
happy.
come on.
I really wish we had been able to get that Chaunax yesterday. That was a cool fish. Oh, there's another fish. Upper right. That one looks a little different.
Push in there.
This is a rat tail.
I'm about to put downlights on.
I wonder if this is similar to one of the ones they collected earlier.
I didn't get to see what they collected earlier.
Jeff says he can't tell which one this one is. Yeah, I'm looking up the images, but
it has kind of like that deeper body with the like really angled to the tail.
yeah, he says it's not as dark as the one they collected earlier, so it must be a little bit different. It's a big sponge off to the...
All right, come wide.
I think we're gonna come up on that coral. Could we look at that as well? Or are we still moving too quick?
No, we can.
Oh, we've got time.
Cool. Let's take a zoom on that. Looks like there's several associates.
Push through the current.
Jeff is happy. Jeff would be happy to try for collecting that fish we saw, but I think it might be. Oh, it's still there.
Is it still there?
Yeah, so if we're ready for a challenge, we can try.
Yeah, sure. Let's try it.
Okay. All right. What's our next slurp chamber open, Taya?
You have slurp chamber 3 open.
Sweet.
And we are on it. Excellent.
Perfect. So Jeff thinks this is probably a Kumba.
Okay, turning suction on. All right.
Which one you want me to touch?
Is it lower cam? Which one's lower cam? I can never remember. If you can find it.
Down below you.
Yeah, it should be right
Can see
underneath
it
you.
right in the upper.
was it.
Very close call. Good effort.
Yeah.
It was a fast one.
Very fast indeed.
That was the closest we're gonna get, I think.
Yeah, no worries. Thank you. Jeff also says thank you.
Okay.
Oh, we can take a look at that Bathy Pathies.
Sure.
Oh, what do we got here?
Oh yeah.
Some Anthomastus tahinotus.
Let's take a look at this. Definitely.
So we've got this Bathy
Push in there.
Pathies with the squat lobster on it. What's that spiky Europtychus? That's so spiky.
Blowing in the wind. Oh. Come on for a second. Lost the footing.
I wonder if the squat lobsters get seasick with all that moving back and forth on the Bathy
All right, you can go back in.
It looks like it's ripping.
Okay, current.
Yeah.
That's good. We can take a look at those really nice Anthomastus tahinotus.
Yeah. In great view right now with the Thurber camera.
Yeah, yeah, that's really nice. Like a little crown on top of the rock.
Exactly. Found its perch.
It's the perfect spot. I got
So-
like 4 of them all right in the same spot. I've never seen them group like that. Usually you just see them solitary.
What else did you want to look at?
The big soft corals.
Okay.
Those are really big.
You can push in there.
Wow. They're really thick.
Yeah.
How fast do they grow? Do you know, Megan?
I don't know. That's a great question. Um, it's hard to date something that doesn't have a skeleton.
Right, definitely.
Um, so we don't really know a lot about their life cycle.
So the fourth one doesn't have a polyp on the end? Or the little...
That one, so these anthomasses can reproduce by budding. So I think that's a new bud.
Oh.
And yeah, it eventually will produce the polyps on the ends and like these other ones. That's great, thank you.
So, what do you think about doing a round of introductions, Blodoc?
Yeah.
Do you want to start us off?
Well, good afternoon, everyone. Apologies for the delay. If you're just joining us live, we are currently around 1,500 meters down. Yes, so my name is Blodoc Kichocho. I am the SCF from 12 to 4. Looking forward for this dive. Yeah, maybe we can just pass it on to you, Meg.
Yeah. So, welcome everyone. My name is May Lubetkin, and I'm the watch lead on the 12 to 4 and the science manager on the ship.
Thanks, May, and good use of the Palauan word Ali, which means hello. Ali, everybody. My name is Else Tale, and I'm the science assisting scientist from 12 to 4 today.
Hi, I'm Taya Jagadas Pollock. I'm just helping out as the data logger for the 12 to 4 shift.
In your front row, sitting in the nav seat, this is Megan Putz.
And this is Jake Bonney in the Herc seat.
This is Sarah Surgeon. I am in the Atalanta seat.
And I'm Erin Rainey, and I'm in the video engineer seat.
Pushing there.
All right, appreciate everyone for their little intros. Ooh,
This looks great.
Yeah, little halocreus jellyfish. This is a pretty
it's
one.
like purple.
Very pretty.
Beautiful.
Sorry, what's the common name?
A jelly. Or you could call it a hydromedusa. All right, we're getting back to the rocks. I love rocks.
Definitely love rocks. So for anybody who's just joining us now, we are around Palau, and we are within Palau's domestic fishing zone just outside the National Marine Sanctuary, diving on a newly mapped seamount. It is unnamed. We just mapped it this morning, and we are heading upslope this seamount to characterize the diversity and distribution and abundance of life in and around these waters. So you'll see us taking lots of zooms. Occasionally we'll be taking small specimens, small samples, to be able to better characterize the biodiversity of this area, and occasionally collecting rocks to understand the geologic setting and be able to date these volcanically originated seamounts. So we're nearing the summit. We're at 1,400 meters at the moment, and we started this dive a little deeper, or around 2,080 meters. So we have a few hundred meters more to go to get to the summit of our seamount. And then on our way up, when we're ascending back to the sea surface, we'll be collecting some water samples as well for our eDNA water sampling program with Pickrick, which Elsa can maybe give a very brief, share with us what you're after.
Oh, there's a fish.
Can push in there.
Oh,
Halosaur.
a halosaur.
That's a bigger one.
It's a lot bigger.
You can see that it has no scales on the top of its head, so this makes it Aldrovandia. I like their little faces. Super cute.
Yeah.
It looks a little like an alligator.
Yeah, a little bit. With that sort of bill nose.
Right.
This one looks very peaceful, very calm.
Very graceful.
Yeah, graceful.
Yeah, up until the point where it gets too close and then it goes zoom.
Is that a nostril in front of the eye?
Yeah, sort of a sensory area.
Wonder what's going on with the coloration of this one.
So this could be Aldrovandia phylacra. It's usually more of a white or gray color, light gray.
I'm very cooperative.
That was a great beauty shot.
That's Jake's great driving.
It's got to be enough zoom for identification there.
If it's not, I can't give you anything else, sorry.
Yeah, well, sometimes
Tough one.
the characters we look for to identify a number of these animals are like internal.
I don't have a lens
So
for that.
yeah.
From video?
Well, with specimens in hand, you
Okay,
might
yeah.
be identifying things based on internal characteristics or things that are hard to count, like fin rays. Or in the case of some really small crustaceans, you're counting the number of serrations on very hard to see parts of like the mouth.
Interesting.
We have a question from the chat asking, what do they eat? If you know.
The halosaur?
Yeah, the halosaur.
Ooh, good question. Megan, what do halosaurs typically eat? What would be their preferred kind of food in this?
I'm not exactly sure what they eat.
Yeah,
I'm assuming they would eat, you know,
It's
small things.
scavenge kind of whatever's
Yeah,
around.
they're not really beta-tending fauna. They're not scavengers. So they don't get attracted to our baited traps.
See. Cool.
Do you mind if I ask about the baited traps, Megan?
Sure. Earlier this year, in March, National Geographic had an expedition out here to Palau where
yeah.
we deployed these baited camera traps where basically it's a camera with some fish attached to it, and that smell of the fish attracts scavengers from around an area. It takes usually a number of hours before we start seeing any animals showing up at the bait, but this is a great way to census some of these more mobile animals that might actively avoid an ROV. We can easily survey corals and sponges that are sessile. They don't move around and cover great distances with an ROV. But for things like sharks and some crabs and amphipods and some shrimps and our rat tail and cut scale fishes, we need to use a different method in order to see what's in the area. And we do that by attracting those animals to our camera using bait.
Ah, interesting.
Appreciate that.
So with different methods, we're able to get the different types of creatures here in the deep sea.
Exactly. Yeah, there's no one way fits all kind of sampling method for understanding the ocean. That's why we use an array of different methods in order to better understand these environments, like using eDNA and collecting rock samples and using ROVs and sampling methods that vary. That way we get a better full picture of what's out here.
All right, thank you, Megan.
Thank you. This is a really pretty one.
Yeah, this is a Calyptrophora.
Quite long.
Yeah, this is great.
Still cam frame up for this.
I am getting photos. I don't know how good they'll be, but
We definitely have improved our photo taking abilities with a good clean of the lens, but still having some focusing challenges, I believe.
Just make a quick shout out to our viewers. We have Jason online. He was saying hi to everyone. Jason from Michigan.
Cool.
And also Devin. Devin was part of the trip from American Samoa to Palau. And yeah, welcome everyone. Thank you for joining us as we continue our trip up this seamount.
Awesome. Hello to both Jason and Devin. Thanks for joining.
Joining from my home state of Michigan.
Do we have any viewers from Palau?
Let me check, please. Oh yeah, we got a couple viewers from Palau. You got a couple of viewers from Canada, Australia. We got Guam. We got New Zealand in the house, Malaysia, United Kingdom, and the USA.
Wow.
Yeah, welcome everybody to Nautilus Live. I hope you guys are enjoying. Please drop in a comment.
You want to zoom that jelly?
Yeah.
And if
You know it.
If you're watching live on YouTube, feel free to go to nautiluslive.org and you can send in your questions. We have a question
push in there.
question portal there, and we're happy to answer anything. Nice.
It's not blooping at the moment.
Oh, and Romania. Hello, Romania. Ooh.
That's cool. I have a few friends in Romania. I wonder if it's them.
Yeah. They were fast to get out of the chat and tell us that. Somebody shouting out the lions.
Right, thanks.
Nice. Great.
Bloodhawk, are you able to see how many Palauan people are tuning in from Palau?
Yeah, we have, it's very nice. It says we have about four.
Oh, okay.
People from Palau, yeah.
Ali Rohoy. I was, before I came on and I was prepping, I was watching the first voyage, so I had it on in the background. I think a lot of people will turn it on and then do some work.
Yeah.
I definitely did the same.
It's a good way to not get work done, though, because I get really distracted if it's on.
I used to listen to it in the background in high school.
Oh, and here you are, leading the science team.
I was a big Nautilus Live nerd from the beginning.
It's really cool what is done on the ship and by the team. Oh, fish. I see a fish in the still cam.
Yeah, in the upper cam.
Yeah.
There it is.
It's another halosaur.
So we have a viewer asking about the great green laser points. I think that is really to give us a little sense of size. So those two green dots is 10 centimeters.
Exactly.
Yep.
Yeah, 10 centimeters.
Scaling lasers.
Yeah. Very helpful as we're zooming in and out on big and small things. It helps us give us a sense of scale.
Hello,
Nice.
halosaur.
Very nice zoom.
It's a little heart shape.
Oh yeah.
All right.
Would this be similar to the other one we got a zoom on as well?
That could be a different species. I think we've got two species here. There's a little bowl of soma.
So speaking of the sizing lasers, I do have sort of a funny story from the first time I was on Nautilus. And I was on a different watch. And we had someone from Hawaii sitting in the Atalanta seat. And if I'm not mistaken, they're responsible for turning the lasers on and off. Or I don't know how it was on our shift. And we actually, on our watch, caught one of those viral moments of a creature that was really interesting, so we were zooming in on it. And you can hear people in the background saying, "Turn off the lasers, Jake." His name was Jake. Jake was on this cruise as well on another shift. And the audio ended up making it into the viral video. And it was so funny because you see all the people in the comments just saying, "Turn off the lasers, Jake, the lasers."
That's always really funny how when you see something really exciting, everyone always just makes such funny comments, whether it's about lasers or just a lot of oohs and ahs.
Yeah.
Yeah, I don't think I've ever had like a good comment during a viral moment.
Yeah, another one of our members of our science team who's off watch, I know his policy was anytime you start to see something amazing to just mute your mic so you don't sound silly in the viral video.
No, I think that's the charm.
It is
Oh,
very
fish.
charming.
Ooh, I see my favorite coral in the still cam.
What's your favorite coral? Is it Iridogorgia?
It's so pretty.
Yeah, it's very pretty.
The firework coral, I do remember the name of it because that was the one I really liked.
Yeah.
Is it firework coral is the common name? Sorry.
Oh, that's the common name that we've made up for it. It's
Okay,
not
our
official
nickname.
or
Yeah.
anything, but
Can we get a zoom on that Iridogorgia
Yeah.
this is
Push it there.
Iridogorgia magnispiralis.
with a little associate, a shrimp associate,
Yeah, this is,
spot lobster as well.
Is this the first one we've seen?
Definitely on this watch. I'm not sure about this cruise.
I think I saw some before, but both cruises kind of run together.
Yeah.
The shrimp is Bathypane.
Turn off the lasers, Jake.
We have our own Jake now. It's so fun. We had 3 Jakes on that cruise.
Yeah, we did.
Yeah. And now we have 3 Dans. 1 Jake, I think.
Yep.
Yeah.
I'll have to text Jake, other Jake. Turn off the lasers.
Yeah, again, so the lasers are not telling us any distance. The laser is just showing us a little size, so we can have a better estimate of what's there by using the lasers to kind of gauge from there, because between the lasers is 10 centimeters. Yeah, we had a viewer that was asking, is it for seeing the range of how far? So it's really just to see the size, have a little.
Come on.
And it's a little tricky because here, when we were seeing the lasers projected onto that rock face, that's what was 10 centimeters, but what we were zooming on was much closer to our depth of field, so the lasers are 10 centimeters to infinity, so whatever they land on, that distance will be 10 centimeters. So if it's way off in the background, you have to kind of imagine that something is going to be quite a bit smaller than their distance, if that makes sense.
Yep. Thank you.
So one of our scientists ashore is sharing some interesting details about that heart-shaped part of the halosaur. Kenneth Sulak is sharing that that's part of that's its brain, and the frontal bones of this fish are really thin and transparent, which allows light to pass through and be sensed by the pineal organ in the middle of the brain. So when it's breeding season, because they actually are seasonal, these halosaurs, the males block off the pineal window, investing the epidermis over the frontal bones with dense melanin to block off light. It's kind of interesting.
Yeah, that's weird.
I was not aware of that. Thanks for sharing, Ken.
That's such a cool part of this, is having experts from all over almost on board.
Yeah.
Another interesting thing that he just shared is that the depth of 1,400 meters, which is approximately where we're at 1,360 meters, is just 200 meters below the absolute limit of solar photons. So what that means is that's the absolute limit that the sun's light can reach down into the deep ocean, which is about 1,200 meters in the clearest ocean waters at the equator at noon, so where the sun can most brightly shine. So it's interesting to think about this fish and its receptivity to that. And only one photon, so one particle of light per square centimeter can actually survive to this depth, which is pretty remarkable. If you kind of imagine light as small particles shining through these deep waters, which ends up being within the range of these fish. About 800 to 1,800 meters depth is where they're swimming in.
So interesting.
Very, very interesting. Thank you so much for sharing.
Yeah, it's really cool to learn how they're able to get around since they don't see in the way that we're able to see.
Definitely.
And all those different adaptations.
Yeah, what we're seeing here is really not what a deep ocean typically looks like. If we were to turn all our screens off and just look at black, that's what the deep ocean would look like. So you can imagine that all the ways that animals get around and feed and know where they are and know how to communicate are really different than what most humans do, who are often reliant on vision to get around.
This seems like a good time too to mention that applications are open for the internships for Nautilus Live. So if you're interested in being an ROV pilot, science, nav, video, SCF, these are all open right now.
Nice. Very good shout out.
Yeah, I actually met Jake, our Herc pilot, when he was still an intern. And when I came on, I was pleasantly surprised to see he's now a full-fledged pilot.
Yeah.
That's
Moving
awesome.
up, moving up.
Exactly. Yeah, a lot of us started as interns, myself is included,
Me too.
10 years ago.
Not too long ago.
No,
Compared to a deep-sea coral, very young.
exactly. Yeah.
I'm going to use that scale now forever.
Yeah.
On my birthday. I'm compared to a deep sea coral. I'm a baby.
Push in there.
Exactly.
This is an Actinernus nobilis, a type of sea anemone.
Well, we saw this before, right?
Yeah,
It's so
yesterday.
pretty. Yeah, my time's way off. I don't know what it was.
Yeah, no, it was yesterday.
Nice.
They're really beautiful.
Let's actually turn the lasers off for just a moment to get some
Jake,
nice
lasers off.
laser-free imagery of this.
Jake, the lasers.
Is it Jake or Sarah who's on lasers?
We got it. Jake got him that time.
Gotta be quick on that.
You got the touch screen.
I got it. I'm cheating.
Yeah, so I think the one we saw before was we were looking at the side, if I remember correctly.
I think so.
And now we can kind of see. Does it have a little a hole in the middle?
I'm good on this whenever you are.
Okay.
Yep. Yeah, that's how it feeds, is my understanding.
Come
This
on.
one is
Yep.
kind of neat how its arms or tentacles are a little bit chaotic.
I'm surprised it doesn't have a food name like everything else.
It's another crinoid. I'm a little surprised not to see more crinoids up on this ledge. It seems like such a opportune spot to perch.
There's a fish,
Yeah.
cuskeel.
Yeah, let's get a zoom on that.
Push in there.
It's not being very cooperative.
No.
We just want to see your face.
It's going to do everything but show us its face.
Yeah. Going into its hole.
Yeah, they do like little holes.
Makes sense. There's lots of nice alcoves to hide and swim around. So there's one other interesting bit that our scientist ashore, Ken Sulak, has shared regarding the halosaur breeding, which I think is pretty cool. He says that the emission of bioluminescent light or blue wavelength light from soft corals in the deep ocean and other invertebrates probably also varies seasonally. So if you combine these in conjunction with the seasonal increase in marine snow, this might also have kind of an impact on signaling, which will help let these fish know when it's time to spawn. So a chain of surface to bottom signals that coordinate this seasonal spawning of these fish. It's really interesting.
Very, very. Thank you to the scientist that shared.
Ooh, let's look at that coral.
Wow. Thanks to all of our scientists ashore who are tuning in. We have 16 folks in the chat, many of which from all over, some of which are from on the ship down in the lounge. But just a little shout out to Aaron Judah, Asako Matsumoto, Chris Kelly, Jonathan Womack, Ken Sulak, Les Watling, Piera Bondi, who was on the ship last expedition, and Steve Oskovich. So
Push in there.
thanks, everyone. This is wonderful. Very big.
Yeah. So this is a hydrocoral, or a hydrozoan that has kind of like a coral-like structure called Solandaria.
Wow.
And then in its branches, we have Sternostylus squat lobster, Actinus scyphia, Venus flytrap anemone. Some amphipods.
Fly trap.
Yep, there's that flytrap. Oh, there's some shrimps too.
Do you know the growth rate for this kind of coral?
I do not. I would assume that the hydrozoans grow a little bit faster than some of the other ones, but I don't actually know. I don't think that this animal has been studied at all. There isn't a lot of stress on hydrozoans as opposed to our octocorals and our black corals, which have been more the focus of these aging and dating studies. We also don't see this animal as frequently as we see the other corals. And the way to tell that this is a hydrozoan is by these super, super fine, tiny polyps. Oh, and there's a tiny sea spider, a pycnogonid in the branches. It blends in really nicely, so you have to look very closely.
All right, come wide.
Can I give you a DVL reset?
Sure.
All right.
That's okay.
Stauropathes, some Pharea. What's that star doing?
Hanging out.
It looks like it's eating something. It might be.
Yeah, let's take a zoom on that.
All right, pushing there.
So this is a sea star in the family Goniasteridae, in the genus Hippasteria. And this posture that it's got reminds me of when it's eating something. So there might be a coral underneath it that it's feeding on, because these animals are coral predators.
So, Taya, you could label this one as coral predation. Great.
All right, thank you.
Okay. Come back wide.
Not sure.
Oh, there's a jelly of some kind in the Atalanta view.
Oh, yeah. It's nice. Speaking of the Atalanta view, Sarah, do you want to share a little bit about what you're kind of keeping an eye on with Atalanta and this kind of eye in the sky of Hercules?
Yeah, primarily with Atalanta, really being aware of where we're at as far as our delta, so the depth above Herc. We want to stay high enough to where we don't get the tether wrapped around Atalanta, but we don't want to be too far to where we're actually pulling Herc backwards or off to the side. So trying to find that balance, but also keeping an eye on the sonar in case there's any large edges coming up, because sometimes Atalanta could have the potential to scrape alongside an edge before Herc does. So kind of a few things to keep an eye on with that. Also routinely surveying all of the pressure systems, hydraulics, power, kind of everything with both vehicles, what's going on, constantly watching gauges. So that way the Herc pilot can kind of, focus solely on piloting, even though he's excellent at
I get distracted
multitasking.
easily.
At multitasking, but also, another big part is watching the cameras, trying to make sure that Herc's always kind of being spotlighted in this, because sometimes, depending on with the ship movement, and also if we're stopping and sampling, looking at things, or even making ascents alongside kind of these steep inclines, it can be a little tricky, and it's very small movements, trying to keep Herc in the forefront of the cameras. So that way everyone out there can kind of see what's going on. It's definitely gets your brain working in a lot of different ways.
Definitely requires a situational awareness using a lot of different factors.
Yeah, it's definitely neat to see, especially when we get some really good shots and can zoom in once in a while to see like when you guys are really looking at like a large coral, for example, like the last one, and you're able to capture that with Herc directly looking at it. It's really kind of a magical scene.
Totally.
Ali and half a day, this is Amanda Dedicatoria, filling in for Blodock in the SCF chair.
Welcome.
Hi,
Welcome.
Amanda.
Welcome. Welcome
Thank
to
you.
the 12 to 4.
This is so fun. So, May, what kind of geological formations are we looking at?
So we are moving upslope on a seamount just offshore Palau in their domestic fishing zone, and it is of volcanic origin. So most of what we're looking at is volcanically originated, and that would be basalt rock covered in this botryoidal manganese oxidized sort of crust. So when we do some nice zooms, you'll see these sort of rounded ball-like textures, which is a mineral habit that
Large fish off to the left to port.
commonly occurs as basalt and other
think it
substrates.
is.
Oh wow, this is
Oh,
really
yeah.
big.
That's a big fish.
Yeah, let's take a look at that.
It's a slickhead.
A slick, this is new then. For us, right?
Yeah, this is new. I bet Jeff's pretty excited to see
This
this.
is really exciting, yeah.
It
I,
was
I don't see a Jeff Drazen is typing, but we will.
I think
Wow,
he's
look at
just
the
in awe
eyes.
right now.
it was like moving so slow in the
Oh no.
Oh,
Yeah, so this fish is the family Alepocephalidae.
and is there a common name for this fish?
Slickhead.
Slickhead.
Wow.
Do you know what it eats, or like how big it can grow up to?
I don't know a ton about these fish, but they do get pretty big. And most fish down here will eat small invertebrates, other small fish. They call it a slickhead because it doesn't have any scales on its head.
And, push in there.
Nice. Let's get the lasers off for a moment as well, since we got some nice scale on it already.
Look at those eyes.
Such beautiful eyes.
Wow. This fish looks very wise.
Right?
It
It's
seems
probably
to have
pretty
been around.
old.
Indeed.
A lot of the deep sea fish live extremely long lives.
Just zooming in here.
It's giving us a great view.
Very cooperative fish.
Did you try to get some pics?
It's beautiful.
Are you happy for me to zoom in a bit more on the head?
Yep, go ahead.
Okay.
Oh yeah. Yeah, we can definitely do that. Yep.
That's
I mean,
amazing.
would you want to do it now?
And you see that little white thing? That is a parasite. It's a naphid amphipod,
Okay, great.
or isopod.
So maybe after we get good
that's
imagery, then
like
we'll
that
do it.
little mosquito, like isopod.
Scales.
Yeah,
What's
those
an
are
amphipod?
beautiful.
This is fantastic.
Isopod is like a roly-poly or pill bug. So there are terrestrial versions of isopods and also a lot of aquatic isopods. And they can range in size greatly from, you know, a few millimeters to like a foot to be like a giant isopod. Though we won't see giant isopods around here.
This fish is well over a meter in length. And I believe it's the first time we've seen one of this size on this expedition, or the previous one as well.
Yeah, I think this is our biggest fish.
This is definitely our biggest fish. So let's keep getting some excellent imagery, and once we're happy with that, we're actually going to fire a Niskin here per Elsie's request to get a good, since we have an extra Niskin bottle.
Okay.
We can keep getting some nice imagery and then do it.
I'm coming up a little bit. You might get a little tug. Just real close to the edge.
Yeah. All right.
Turn my auto heading off for a moment.
Yeah, you can
I'm
just
going to
come
go
up.
wide,
Okay.
just so you know.
Jeff is being facetious and saying that we should collect it, which would be completely impossible.
I'm fully wide
Where
now,
would we
Jake.
put it? It wouldn't even fit in the box.
I wouldn't even want to. This fish is...
It's probably quite old, right? Is that
It's-
what
Oh
you
yeah,
said?
it's gotta be very old.
It's holding the knowledge of the seamount.
That was gorgeous. Those are some great shots. Nice flying.
Yeah, Jake, great flying.
Fish did all the work.
I always find that I end up about 2 centimeters from the screen trying to... That can't be good for you.
All right, so...
Well, I'm always like leaning over. By the end of the watch, I have like a crick in my back.
I know, I keep
Just
stretching.
go on a Niskin.
Yeah, let's go for it. Let's go for a Niskin. I believe that's Niskin number 2, right? Is that right, Taya? Yes?
Yes, that's right.
Awesome. Fantastic.
Drag date. Pan left.
Tilt up a little bit.
Elsa, do you want to share a little bit about why we want to trigger this extra Niskin bottle at this location and the interest in fish?
Sure, thanks, May. So we are collecting environmental DNA, or eDNA for short, using the Niskin bottles, which are handily on Herc. We have 6 of them. We are doing them at predetermined depths starting from the bottom, and we had 1 extra because of the shallower depth. And we wanted to prioritize vertebrae, so fish, whales, other things which are usually mobile besides benthic organisms.
Would you like the lasers back on?
Sure.
Because the purpose of our eDNA project we're currently working on in partnership with the Stanford Center for Ocean Solutions and of course the Ocean Exploration Trust is focused on fishery. So we are interested in sort of getting a picture from not just spatially across the surface and also in the depths.
Awesome.
So thank you, Jake. Thank you, Herc, for that collection. This morning we had collection by Mike, so thanks, Mike, over the side of Nautilus.
There's a sponge.
We definitely would have needed a bigger net.
Yeah.
That was a big fish. Thought it was a rock at first.
Could just barely see the movement. It was more the shape.
Was that you who spotted it, sir? Nice. You're becoming quite the fish spotter.
I do enjoy fishing. This is just a different kind.
Can we do a little zoom on that grayish, brownish one? This is a type of black coral.
All right, push in there.
I wanted to get a good look at how it's branching.
Why are they called black coral?
Because the skeleton is black, but the tissue can come in many colors, from white, transparent, to bright red and orange.
We had that dead one yesterday, right? And you could see just the skeleton, the black skeleton.
Yeah. You can kind of see the black skeleton through the tissue here. I believe this is a heteropathes. Thank you.
Okay.
Yep.
Bounce.
All right, so I think given the amount of time that we still have left, as we come up here on waypoint 5, we're, instead of going straight up to the summit, we'll take a little bit of a lateral turn around the seamount.
Okay. Do we want to follow a depth contour, or
Yeah, I'm thinking which direction to go. I think probably going clockwise.
okay?
And okay, Brian just agreed to go clockwise as well. And I think we can sort of come up, sort of follow one of those contours, but slightly be heading upwards, like a gentle spiral, if you know what I mean.
Okay. Yeah, just like every few
Doesn't have to-
meters move up a
Yeah,
depth contour.
sure, yeah. So if you want to go a little bit further than that, like,
So like this is 75 meters that I
Yeah.
have. So like every 75 meters move up
yeah.
around and spiral up to the top.
Yeah, we could even go a little bit slower than that, so go a little bit more, follow the contour a little bit longer, just
Okay.
given the amount of time. And
Yeah,
then
we've got a lot of time.
if we end up running out, we can always just beeline it to the waypoint 6 from wherever we're at.
Okay.
You're telling me the DP doesn't have a spiral mode?
Yeah.
It'll be a nice challenge for the coordination of all vehicles.
So we have a question from one of our viewers. Else, are there any local stories or connections to this particular area of Palau that you know of?
Thanks for that question, Amanda. Yeah, it's pretty interesting in that the sort of migration of ancient Palauans, which originated from actually an area called Australasia around Indonesia. And then they actually migrated to Guam first, and then out towards the east, or south to Palau. And once they got there, after the voyaging, we actually found a beautiful set of islands which had everything that we needed right in our reefs. So to my knowledge, there has not been any ancestral migration out to the deep sea besides the original voyaging that we brought back. But that's sort of the really interesting thing, is that now with modern technology, we're able to bring Palauans on board to come to see what's out here in our ocean, and then also stream it live for people watching at home to see. And it's really interesting, and I think a lot of Palauans who have the opportunity to either see this live or to see the media around it will gain a really great appreciation for what is in our deep ocean.
That's wonderful. Thank you so much for sharing.
Yeah, we have a whole plethora of connections to shallow water species. We have very specific names for a lot of different fish, and it's very well covered and well studied. And now we're venturing out into the deep sea. So yeah, all of the seamounts are unnamed. So I don't know if anyone knows the process of naming a seamount. Maybe you could.
The process of officially naming seamounts and species is one that is quite lengthy, and
Okay.
We could use the word bureaucratic as well.
I see.
But definitely, especially in the last several years, there's been significant effort put towards focusing on culturally appropriate and relevant naming of deep sea inhabitants and species that are newly encountered by humans, and as well as seamounts. So I don't believe that's part of our plan to work on naming these seamounts, but
I want to assume that crinoid.
But definitely, if they were to be named, they should be named by those in Palau who want to extend their stories to the deep.
Push in there.
This is a crinoid called Pentametricrinus.
Nice.
Nice.
Cool, thank you.
Okay.
It's a pretty one.
Come back, go ahead.
Just thought we should see it.
And
That was great.
actually, our chief scientist, OET's chief scientist and expedition leader, Daniel Wagner, has named several seamounts in the past, so he may be able to help our friends in Palau with naming these features, if that's of interest.
I'm sure it might be, so we can, and even though it might not be a part of the work of this voyage, I think we've definitely started that process just by mapping them.
Definitely.
As you said, this is previously unmapped, unseen by human eyes, and here we are.
Absolutely. it is definitely helpful when seafloor features or areas are named, because otherwise often what happens in the scientific literature is a lot of different people will have their own names for different things, and we might be speaking about the same thing. So yeah, it is great to be able to actually call something by its official and culturally appropriate name.
Yeah, and I think in general, I think names and are sort of in science, it's very fluid, or in terms of like species and changing the taxonomy, and we recategorize the species, and this might have been called this before, and now it's something else. So those are things that we run into when we do our work in the shallow water, where we'll be identifying especially corals, and come to find that they've changed the name in the past 5 years.
That happens a lot in the deep sea too.
Yeah.
So there are some things where I'll call it a name and it will have been called something else previously, or I'll call it the old name, and somebody will be like, oh, it's called this now. I'm like, yeah, no, sorry.
Science is definitely a process of observation and iteration and revision. So using what we understand when we observe things and the evidence that we have with what we currently know, our knowledge can adapt and change when we learn more things, and that's how we revise our definitions or our categories of various features and organisms. It's a very ongoing process and a dialogue with all the previous scientists and observers and knowledge holders from the past.
Exactly.
Is that another cookie star?
Yep, probably Sarah Master.
Seen 3 of those in the last few minutes. There's another shrimp.
2 shrimp.
2 shrimp. Oh, yeah.
Ali, and welcome to our viewers just tuning in. We are currently exploring an unnamed seamount west of Babeldaob Island, just outside the boundaries of the Palau National Marine Sanctuary. This is a 16-hour dive with goals to survey the diversity and distribution of deep-sea benthic communities and look at the area's geological features to better understand the age and volcanic history of the region. If you have any comments or questions, please send them over on nautiluslive.org.
I have a question, May. So when we talk about dating the rocks, could you talk a little bit about how long we could expect that to take?
Ooh, that's a very good question. So I'm not actually sure if we have a scientist ashore who is sort of lined up and planning to do these analyses.
Hmm.
I'm not sure if we have someone. If we did, the way that that process would go is our samples are sent to essentially sample libraries. So all of the eDNA samples
Ooh,
are,
that's good fish.
Push in there.
yeah, let's get a nice zoom of that.
This is a different fish. We haven't seen this fish before.
Ooh, new
See, it
fish.
has a forked tail. I think it's in the same family, the slickheads, the Lepocephalidae. Like the big fish that we just looked at, but this is little. If we could slurp this, I bet Jeff will want it.
Yeah, Jeff says it could be a Melamphidae.
My light's still on? No.
It's just really dark. I've got...
Yeah, no, it's really fast.
Yeah.
Oh, there's no way we're catching that fish.
Oh.
Do you want me to come wide?
Hey, come on.
There's another shrimp. But yeah, as I was saying, so our samples that are collected on this expedition will go to our partners at PICRIG, the Palau International Coral Reef Center, for all of our eDNA samples. So all of those water samples that have been filtered by Elsa and Greta tirelessly throughout our days and nights. But then all of our geological samples and biological samples will be heading off to 2 other sample repositories or sample libraries that are facilities well equipped for receiving these types of specimens, and not only receiving and holding them, but it's okay, yeah, we can look at that. But also sharing them with any scientist who's interested in studying them. So they work as sort of lending libraries. So scientists around the world can check out a sample and do some research on it and either send it back or use just a part of it. So
What do you want to look at?
do you think it's an urchin?
Sorry, or I guess I'm just wondering what...
I see.
Oh, yeah.
Cucumber?
Yeah, we
It's
can.
a urchin underneath that ledge.
Ah,
Yeah,
okay.
so looks spiky.
Push in there.
And we've got a...
Yep.
Is that a Farreid?
It is.
It's a little one. Little Farreid's glass sponge to the right.
And there's a couple cup corals as well.
Right here? Is that an
And
anemone?
there's one
Yeah,
here.
that's a cup coral.
Ah, okay, nice. Yeah.
I'm sorry, what was the common name for that urchin?
A pancake urchin.
Got it, thank you.
Thanks, Jake.
Yep.
And yeah, thanks, me.
Yeah, so once our geological samples reach the Marine Geological Samples Lab back in Rhode Island, then an interested scientist will be able to request those samples and do age dating on them. So that process will involve most likely carbon dating, and depending on whether that researcher is doing it in their own lab with their own settings or sending it out to be done, it can take Probably from now until getting some answers, I would say several months.
Okay.
But depending on whether or not
Oh,
there's a
that's a bat
researcher
sponge.
planning to do it right away, or if it's more a collection to have for future studies, might take longer.
Yeah, that makes sense. And I think since we're already here exploring, mapping them, you know, is an important first step, and
Push
then
in
getting
there.
the rocks just to have them
Exactly.
in our repository or library, I think you called
Yeah,
it.
both work. Yeah.
Yeah.
Yeah. So generally speaking, we like to leave these environments intact and only sample the smallest amount that we need to
Do
characterize
you have
the
more zoom?
biodiversity and geologic
Yes,
setting.
I do.
So we take small snips or a single rock here and there in order to map these habitats in situ.
That's great, thank you.
All right, come on.
I'm going to
Max?
guess that this is crunchy.
It is crunchy.
It is crunchy. Good guess.
Yeah, this is a sponge in the family Eurydidae called Conolasma.
Nice. Is that another one?
Yep.
Looks really similar. Some cookie stars. Ooh,
There's a
several.
lot of cookies.
Yeah,
So many cookies.
5 cookies.
I could go for some cookies.
Yeah.
Yeah.
I see even more cookies in the upper cam.
It's cookie station.
Yeah. Getting into
It's almost cookie time on the ship.
Yeah,
3
it's
o'clock.
another hour for cookie time.
What's your favorite cookie?
Oh, chocolate chip, absolutely.
Yeah?
Ooh, that's a good question.
I have some in my bunk, so hit me up if you need any snacks.
Ooh.
It's an open offer.
Nice.
Watch out what you offer.
So is this very wide angle view the camera at the very top of Herc?
Uh-huh. Yeah, it's like a fisheye view from the top looking down.
Got a
Looks really cool.
fish just below you.
Oh, I do see it too.
Looks like a halosaur.
I think you're right. You're right.
Yay.
Several halosaurs on this dive.
Yeah, that's Halosaurus. It had scales on the tops of its nose.
Did you say Halosaurus?
Halosaurus.
Is that the
That's the genus.
genus?
So for all of our viewers and scientists ashore, we are moving up along a seamount near Palau in Palau's domestic fishing zone, just outside the marine sanctuary. We're nearing the summit and doing a little spiral around laterally. We're just past waypoint 5 for those following our dive plan. Surveying this upper part of the seamount.
I had to run out for a sec. Did you figure out what that last little fish was? The really dark one?
Oh, did I figure it out?
Yeah. Or was it figured out?
I think Jeff had an idea of what it was.
Sorry, what?
The small blackfish.
Yeah, that was, well, there's two possibilities that we've been proposed. Maybe the family Melamphidae, or possibly Stephanobarax. That dark one is likely in that family of Melamphidae. That's what Jeff says.
Cool.
So our SCF, our 12 to 4 SCF Bullock is back from an interaction. Many thanks to Amanda for joining us for a bit.
Yes, thank you, Amanda. And I'm back.
Was that with Palau School, your interaction?
Yeah, it was one
cool.
of the elementary schools, and it was just had a lot of questions about what's going on. And yeah, it was pretty fun. Young kids, curious, wanting to see what's the worst we've seen.
Awesome.
Yeah.
That's really cool.
Yeah, they were also asking about sharks, so hopefully we get to see some during our dives.
That would be very cool.
In school, do you mostly speak Palauan or do you speak English?
Like when we did
Push in there.
we tried to speak mostly Palawan, but some of the students were like talking to us in English, so we'd respond to in English. But in school, it's really has changed because of maybe technology. Students now are more used to English than they are Palawan. So a lot of things is we try to utilize Palawan as much as we can, have them practice. Wow, that is so interesting. What is that? Jellyfish?
Yes.
Jelly.
Too close.
Come on.
So your interaction was in Palawan?
Yeah, yeah.
Okay.
And it was, I think they were like third graders.
Oh, that's so.
That's fun.
Yeah.
That's so fun. For our viewers out there, do you want to share just a little bit about what an interaction is?
Okay, so interaction, if you go down to our website, you can find an area where you can sign up for these interactions, where the SCF and some of the members from the teams come out and talk about the mission, answer some questions.
Jake, can we look at this coral?
Sure.
Yeah, and I had a couple interactions last night with some schools in the U.S. We had a Lego. They had a team that asked for interaction where we were talking about
Push in there.
robots.
You were up quite a bit last night doing them.
Yeah. They were thinking about making.
This one's interesting.
Very bushy.
Mm-hmm. Will we be down to collect this?
Yeah, we could make a snip of one of these. In terms of biobox space, we have those little ones, but we also have that floaty holothurian, right? That floaty sea
Yeah,
cucumber.
I wouldn't want to open that. Also, the black corals tend to be very floaty as well, so
Yeah.
I'd rather
Slurp it.
slurp it.
Yeah,
Yeah.
if it's not too wide, and
I
we
was
just
gonna
take a small snip, then
Okay.
That sea cucumber already tried to make an escape.
yeah. And our forward biobox is off limits too. It's got some escape potential.
Escape artists on board.
Yeah, exactly. So would this be slurp number 4, Taya?
No, this is slurp number 3.
Slurp number 3? Okay, cool. That's right. We didn't succeed with that fish before.
Almost.
Yeah.
Is that good, Jake?
Yeah, that's good.
Okay.
And try to get it really nicely on the coral cutters. These can be hard to cut.
Okay.
Can
All right.
probably
That looks good, yeah.
nice.
Oh.
Nice.
All right, come forward.
Get another screen grab of that, Taya.